are genetically edited people walking among us right now. Could somebody be in a seedy clinic somewhere getting their eye color or their hair color edited? What about designer babies? Are those a thing yet? And how come we're not just gene editing cancer out of the population? Well, maybe we are. I have some answers for you, so let's dive in. Answer number 1 is yes, there are genetically edited people walking among us right now. That's the quick answer, but there's more to it. As you might imagine, it is a very small proportion of our population, but who knows? It could be your neighbor, it could be your coworker. We're talking about people who have either participated in an above-board clinical trial or people with certain blood disorders like sickle cell disease who have used a government-approved gene editing medicine called Casgevy. Now with Casgevy, it's not like you're just taking a pill or getting an injection and having those edits done inside of your body. That's not what we're doing. In the protocol for Casgevy, you need to take the diseased cells that you want to edit. So in the case of sickle cell disease, it's your blood stem cells. You take them out of the body, you edit them in a dish, and then you put them back into the patient. Why? To reduce complications. When it comes to the edits themselves, there could be mistakes, and we call those mistakes off-target edits. These mistakes could include anything from just cutting in the wrong place on the DNA strand to editing the wrong type of cell if this were to just be dumped into the body without safeguards. So think wanting to edit the skin, and then you actually end up editing the muscle instead. That could cause serious health issues, including tumor formation, so we want to stay away from that. Scientists are also working on ways to send the editing tools to very specific diseased parts of the body. It's almost like giving them a mailing address. Theoretically, that would also minimize those off-target mistakes, but again, all of that is still in R&D. In the case of Casgevy, CRISPR is literally just zapped into the cells. These are just specialized electrical pulses that'll create tiny pores in the cell's membrane, just big enough for CRISPR to get inside of the cell. Now, the current standard of care for injectable medicines that involve the delivery of a payload usually requires the use of one of two things. So it's either going to be a special virus that's not going to make you sick. It's basically just like an Amazon package, really. They're not even able to replicate anymore. They've been stripped of all of those components. The second strategy involves something that the vast majority of us have already been exposed to. I'm talking about lipid nanoparticles, and these were used for the COVID vaccines. However, both of these are still foreign particles that are entering your body, and so they can still cause problems, especially when it comes to your immune system or your liver. Are we making amazing progress? Yes, definitely. And we're almost there. We're so close, but we're not quite there yet. So for now, editing them in a dish and then putting them back, that is the safest option that we have. I do just want to reiterate though that there are clinical trials ongoing looking at in vivo edits or edits that are done inside of your body, so don't rule that out just yet. I also promised you info on gene editing for cancer, and again, the answer is yes. Scientists are working on this right now. In fact, we have a somewhat similar approach to Casgevy, where a special type of immune cell is being taken out of the body, upgraded so that it can hunt and kill cancer cells, and then return to the patient. That's been approved, and it's happening in clinics at this moment. Based on what I've seen, it is likely to be one of the most promising genetic strategies for cancer to date, but it does have its limitations. It's called CAR T-cell therapy. Super cool. I'm going to be doing an entire video on this topic separately. So think of your immune system as an army. I'm going to take one of your best fighters, the T-cell, and I'm going to equip it with a special sort of cancer facial recognition software that's going to allow it to detect certain types of cancers. I can then put that upgraded T-cell back into the patient's body and watch it hunt and kill. Direct gene edits to cancer cells, that's a little bit more complicated. Cancer is notoriously multifaceted and complicated when it comes to the genetic drivers of the disease. There are those big, highly correlated genes, very recognizable, such as BRCA1 and 2 for breast cancer, right? But then there are other sneakier mutations and genetic elements that can combine and cause a perfect storm. These are much tougher to target one by one, especially considering how unstable they are and how quickly they continue to mutate. But it's not impossible. People are working on this problem right now, and they're even approaching it in ways that you might not expect, like trying to create edits that leave the cancer cell more susceptible to certain drugs, but we're not quite there yet. Now, medical settings aside, okay, we could still have people doing gene edits in more of an underground capacity, even though that would be very much illegal, especially if those gene edits were to affect the next generation. You might have heard about the CRISPR babies controversy from a few years back. This was a real thing, so a scientist actually illegally gene-edited embryos to try and make them resistant to HIV. He put those embryos in the mother had the babies and their children now. Why HIV? Well, his claim was that there was a ton of supporting evidence and research on the mutation in question, and that ultimately just gave him too much confidence to take this on. Even he admits that he did the edits too soon. These kids are probably 6 to 7 years old now, and while we do want to be sensitive to the family, I think the general consensus within the community is that it was both risky and irresponsible to do what he did at the time that he did it. The lack of oversight and the lack of regulation on a secret project like that, those are some of the worst parts. Speaking of which, you also have people using DIY CRISPR kits, and yes, that idea is as bad as it sounds. Thankfully, the FDA is all over it. Please do not try to edit your own genes. Just to wrap this video up, in these very early stages, when you're working with an intervention as risky and as permanent as gene editing, you want to consider what I call the 2 S's. So that's both the simplicity, but also the seriousness of a disease or condition in question. Sickle cell's cause is relatively simple. 1 gene, and that already sets it apart from cancer in many ways. We call this a monogenic disease. Sickle cell is also serious enough that for many patients, the potential benefits outweigh the risks. So there goes eye color, right there, out the window. The ethics of the eye color thing alone. Like, I haven't even gotten into eugenics here, you guys. We are at the very tip of the iceberg. As we learn more and as we advance the science, you're going to see it for yourself. We're going to be tackling more and more complicated diseases and potentially less serious conditions. And just feel like that latter part is not necessarily a good thing. Anyway, you're now up to date on gene editing. If you learned something, share this video with a friend.